Hello, and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines. People just like you working to understand viruses and how they affect you. While our podcast has focused on researchers involved in coronavirus or COVID-19 related research over the past year, moving forward, we will also be interviewing researchers studying other viruses so that you can learn who they are and what they do. I am Larissa Thackeray, and I'm hosting this podcast from America's Heartland in St. Louis, Missouri. The use of oncolytic viruses to target tumors is a field with a long history. On February 9th, 2021, we talked with Dr. Sharmila Nair, a staff scientist in the Diamond Lab at Washington University School of Medicine in St. Louis, about her work using Zika virus as a therapy for glioblastoma, a fatal human cancer. Sharmila received her PhD from the Technical University of Braunschweig in Germany, where she focused on understanding innate immune control of virus infection. In the Diamond Lab, she has continued to focus on the role of innate immunity during infection with a variety of pathogens, including flaviviruses, alphaviruses, and coronaviruses, as well as tuberculosis. Hi, Sharmila. I'm happy to have you with us today. Why don't you tell us a little bit about yourself? How did you become interested in virology research?
I always inclined towards the biology field, and I'd pursued my engineering in biotechnology, it was an engineering degree, but towards biotechnology. And around that time, there was a epidemic that happened in India, which is dengue. Dengue was a big problem at that time, and it went to a point that my university had shut down for a couple of days just to handle. And you hear about all these infectious agents that roam around in India, but like this is the first time where things came to a standstill, at least for a couple of days at least. So that just, since I was in the field, just kind of piqued my interest, started reading some papers about it. I had identified a few groups around the world in my limited reading capacity. But there were a few groups in Singapore, in England, of course, some in the United States, many in the United States, and Australia. Prior to that, I had, done some training abroad during my vacation time. I had an internship opportunity when I went abroad. So I had started to get into learning the techniques of how to, ask questions and answer questions and what experimental questions. And so I started interacting with a few of these PIs, at least whoever would reply back. That's always a thing. Not everybody does reply. But a few of them did. That was it was very kind of them to take interest or take time to even reply to how old was I? 18, 19-year-old. And then just started writing, and then I started to think about doing a master's in virology in a dengue lab. So that's how it started. And then I'd moved abroad to England in the group of Andrew Davidson. He does a lot of reverse genetics with dengue. He's quite a known person in the dengue field, and just started there basically.
Okay, cool. And had you had exposure to sort of biological sciences or microbiology earlier on, like in high school or before then?
Not outside your textbooks. So let me think. No, so I don't think I did much during my my school years. I definitely started getting, so we really get to genetics and anatomy and basic biology only in your 11th and 12th. I don't know what the equivalent in the US is, but It's 11th and 12th. And that always was amazing, just genetics, Mendelian. I just remember being so excited by it. And I think that's something definitely that came naturally to me as well. So I knew I wanted to pursue something in biology. What was still to be determined. I knew I wanted to do engineering only for the reason that I knew I wanted to be able to go abroad and learn more. And I don't know if you know this, but at least at that time, I don't know if it's the same case now, but you needed 16 years of education to be able to go for a higher education degree abroad. And in India, your basic BSc, which would be your undergraduate degree would be a 3-year course, whereas an engineering course is 4. So I wanted to do engineering, but that was hard because the first couple of years you're doing you're doing all engineering. So there was a lot of physics and a lot of electrical engineering and all of that I had to do, but it was good. Not that I remember any of it now, but that was, yeah. But yeah, so going back to the question, but during my college years, I definitely started looking for opportunities and creating opportunities for myself in some ways. So I had done a lot of internships, many of them in companies. I actually did a small internship in Dr. Reddy Laboratories, Bharat Biotech. You must have heard of that because I think one of the vaccines from our lab is now there, being tested there. Then and then I did a few other internships, and then I actually wrote to people abroad, and then I got selected for this undergraduate research fellowship. I think that changed my eyes completely. So I moved abroad for about I think it was 3.5 months. Did some really nice, it was a really nice exposure that I had. Came back, and that's when the dengue epidemic happened, and then and everything followed from there.
Right. And so you mentioned that you went to England, and didn't you also go to Germany as well? So was that afterwards?
It was after. So in Europe, well, first and foremost, I had done my master's first. But in Europe, in general, you need to have a master degree to do your PhD. You can't do it directly from your bachelor's. I think in England it's different, but in Europe in general, you need a master's degree.
So how did you, I guess, can you describe a little bit more detail? So how did you then get to sort of like your PhD and then your postdoc? What steps in a way did you go through? How did you identify those particular jobs?
So the experience in England was really nice. It got me, working hands-on with certain molecular techniques. It was very basic virology, and it was interesting, but not interesting enough for me to pursue my future. And so I started really thinking about what is it that I wanted to do in science. And at least at that time, the two parts that seemed pretty obvious to me was immunity to viruses, or going more towards the structure side, and then thinking about drugs and how you can model and look at things that block and yada yada. And I was really interested in innate immunity, just reading more about immunity. Innate immunity is just sort of like something that was really engaging me. But regardless, I'd actually applied to programs across Europe at that time in both immunology programs as well as I think a couple of even the other programs which had more rotational programs in structural and immunology. So there was one. So I'd applied to a few. I can't remember, but I think there was one something called an Erasmus program, which basically you hop between labs across Europe, almost like a rotation project, and then decide what you like. But before I could get to that, I actually secured a position at the Helmholtz Institute of Infection Research. It was a really great place. It was a government funded. It's like the NIH in a way of Germany, and the project was really interesting, and so I just joined there.
And then how did you, once you had finished that, how did you then think about, how to get your postdoc or where to go, what lab to get to?
Yeah, I would say that the whole process is actually very simple-minded, I would say. So my PhD in Germany was actually really short. I finished the whole thing within 3 years. I published, I did everything. I think by the time I graduated, it was about 3.5 years, but I pretty much was done in 3 years. And then at that time, I had worked on neurotropic viruses, innate immune responses to neurotropic viruses. And at that time, I was just looking at, okay, how much I want to know more about immunity in the brain and potentially basically antiviral immunity at that time. And of course, Mike was a leader in that field. I remember that distinct Nature Medicine paper that had come out was, I think, Hylam's paper that had come out. And So I wrote to him, and he was, of course, the first to reply, which is no surprise. Got the response within seconds at 2 a.m. God knows how. Anyway, it just worked well because he seemed responsive, I was responsive, and it just felt like, okay, this is someone that is engaging, and it just happened from there, and I secured a grant to move to the US, and so that was really nice actually, to secure the grant and move to the US and start my postdoctoral work here.
Right. So I know you've actually worked on a couple of different aspects of virology and immunology in the Diamond Lab. Can you take us through some of them? And then in particular, I'd like you to talk about sort of the Zika virus cancer stuff, but why don't we put that at the end? Why don't you talk about some of the projects you worked on before that?
Right. So when I moved to the US, my goal was to investigate immune responses to infectious agents. So I really like virology and viruses and infectious agents, but I was actually more curious about what really took me was the immune responses aspect. So to some extent, I would use these infectious agents almost as like a tool to understand the immune responses. And all the infectious agents that I actually worked on were actually pretty endemic in where I grew up. So I've worked on flaviviruses. Of course, it's one of the heartlands of India is one of the heartlands of that. And then TB, which is the curse in India, and chikungunya, which is also very, very prevalent. I mean, I can tell you 10 people who have been infected by all of them. That's just the sheer burden of where I grew up. So these are, I guess in hindsight, maybe I could have streamlined a little bit more, but I was just so interested in science. And I'd also just done a PhD for 3 years that I just wanted to explore more and more and more. So anyway, so I initially, when I secured the grant, my goal was to investigate the role of this particular gene called immune responsive gene 1, which is involved, which is an interferon stimulated gene, but it has an enzymatic activity and decarboxylates a crucial intermediate in the TCA cycle, in the citric acid cycle, called cisaconate to a metabolic product called itaconate. And what we found was this product, itaconate, has many immunological functions. So basically, it was this immune metabolic axis that this particular gene was modulating. And I tested this gene, the function of this gene, against a bunch of viruses, but what really stood out was its role in TB, and at that time it just felt like this is something I had to do. And so I pursued that, and we found some really interesting data, which we published almost 2 years back. And then I was also involved in projects with chikungunya virus and antiviral roles in stromal cells. So when I'm particularly interested in unique antiviral signatures in specific cell types. And what I had found was the role of interferon regulatory factor 1 in having a particular role in specifically muscle cells, but not other stromal cells, in controlling viral replication, which is crucial because it's one of the major targets of replication for chikungunya. So we published that too a couple of years ago. And then moving on, I started investigating the role of Zika virus in modulating, in basically working as an oncolytic agent. And so there have been studies which were published which showed that Zika causes microcephaly, which basically the question arose was, why is it that Zika is not really infecting the adult brain, but it is easily infecting, fetal brains. And one of the hypotheses was, well, there are a lot more stem cells in a fetus as opposed to an adult. And this is something that subsequently was proven, at least in the mouse model, was that Zika can really, actually not in the mouse model, in culture and in human biopsy samples, that Zika can specifically target to infect and kill neuronal stem cells, which is a big, which was later put into therapeutic use. At least that's the goal, because in many tumors there are cancer stem cells that are generated, especially in glioblastoma, which there is no treatment for today other than standard care. And even with that, your chance of survival is basically, your basic median lifespan is 6 to 2 years. And once you diagnose, death is pretty much inevitable even today. So the question of using Zika as a therapeutic agent really came up, and I was really happy to contribute to that story.
So can you tell us a little bit more details about this? So I guess just a little bit of the backstory, like this whole idea of using viruses as an anti-cancer therapy, particularly in the brain, sort of has a long history. And, and then also, I guess, some of your studies where now you're actually starting to look at what is once again, the immune component of that response.
Right.
Tell us a little bit about both of those.
Yeah, so I think it's interesting because the earliest accounts that I can find of using infectious agents for therapeutic purposes has been dated as far as, I think, the 16th century, where people, 16th or 18th century, I might be missing something there. But anyway, the point is, people who had, I think, leukemia and also acquired herpes virus infections, they went into remission temporarily, and then of course it came back, and then of course these people died. But these things have been documented in history, and I think in the beginning of the 20th century is when people really started doing clinical trials. And today you would question their the ethics behind those trials. But people started just throwing viruses, like all viruses, through West Nile, flu, and herpes, and I don't know, and just saw whatever sticks. And then in the 60s, it went down again because I think, a lot of the agencies for ethics and stuff really started coming up. But, people have still followed through all the way to the 90s. And it's really nice now because now there is an FDA-approved oncolytic virus, which is the herpes virus for the treatment of really advanced melanomas. So that has really, again, given hope to people who are in the oncolytic viral field to pursue and basically look at better, bigger, better, more potent viruses that can specifically target, tumors without much toxicity to the humans.
Right. So how does it work for Zika? So when the Zika goes into the tumor, what is your work shown? What is happening essentially?
So I think it's a two-step thing. It's doing two things, and that's the advantage of Zika over other viruses. One of it is that it's specifically targeting the stem cell population, which is the population which causes most problems in resurgence. It's that population that is very hard to irradiate. It's very hard to get rid of. And then what the second thing it's doing is it's actually evoking an immunological response, especially a CD8 T cell response. It's not only promoting better numbers to get recruited into the tumor, but also improving the functional responses of the CD8 T cells against the tumor into the tumor. So I think it's a two thing. I think that's why it's superior. But of course, we need to do. It's a long road ahead before it starts going into a therapy in a therapeutic setting.
Yeah, of Cool. So I guess more generally, I have a couple questions about, so what has been the most exciting moment in your career so far?
It's incremental for me too. If I find a day that's absolutely exciting, that will make my day for that moment. And then you move on and then you find the next thing. But also sometimes when you get sudden validations, like when you get some awards, you get grants, that makes me really happy because you know you're doing something right in life. Um, um, but I don't think I've had that eureka moment. I mean, if I get a patent which is out to the world and I make $1 million, of course, that'll be the eureka moment, but I don't think that's happening anytime soon. So maybe my bar is too high.
So I guess then maybe conversely, what's been the most difficult thing you've had to overcome as a scientist so far, and how did you overcome it?
I think it's, I don't know if I've overcome it. Let me just put up there. It's a work in progress. But it's for me, it's like the balance between passion of what you really like to do, but also staying on track to whatever defines your success path. Like, when people say, do what you love. The problem is I love too many things. I mean, in science, like I really love immune responses to this, but I also really like to investigate that. And I mean, if you're a PI, maybe, and with a well-funded lab with multiple grants, and that's something that you can do. But I think as a postdoc, one has to be really mindful of your time and what your targets are. And although it feels like you're letting go of some things, maybe that is the case. You are letting go of some things, and maybe you would have had better success there. And there's no way to know till you try. But getting that balance right is important, I guess.
Yeah. And then I guess if you had a chance to ask your older self, say you 60, 70, heading towards retirement, one question, what would it be? What would you want to know?
I would ask, what would I do differently in my 30s? Because I'll do that now. But I think, more serious note, I would really do that if that was a possibility. But I think I would say, were the sacrifices worth it? I think that would be my major questions. Are you happy with your life, and were the sacrifices worth it? Because I have left my family. I have left my family for the last 10 years. Not that I don't see them, I see them very often, but I miss home. I miss home a lot. So so I think I would just hope that I've been happy with my life, I suppose.
Yeah. So I guess for this, obviously everyone's life has been changed in the last year due to the COVID pandemic. How has it affected you as an individual? What's changed? How has it affected you?
To be honest, I cannot separate what's happened with the pandemic with the reality of my life, in the sense that I'm also a new mother. So it's been chaotic generally. And I really don't know how much of it is actually attributed by the pandemic. Maybe some of it, maybe none of it. I really can't tell. My son is about 13 months old now. So he was just about 3 months when it was the height of the pandemic, which is pretty chaotic time for me anyway. So I really can't tell.
So how have you dealt with things like childcare and safety? I think it's a different, for a lot of our listeners, it's one thing being an adult or, dealing with your grandparents, but how do you deal with it as sort of a new parent?
Yeah, so this was something I stressed a lot about initially, because initially the plan was my mom would come and help me for the first 6 months, and then we were fretting about what we were going to do. And I was really scared about sending him to daycare because my kid was also born prematurely, and that was weighing in and causing a lot of stress in my life. And I mean, thanks to my family, they really stepped up, and somebody else in my family just came and stayed with us for the remaining of after my mom left. So that really was a big relief. But I also realized that my situation is not the same as a lot of the other, new mothers who had children around the pandemic time, and they've had to battle the same questions and had made different decisions. But I really feel for all of them, because it was not an easy decision easily.
Yeah. So, and then I guess to follow up on that, so how do you make decisions about sort of, going out for shopping or, now, I assume you've been offered the vaccine through WashU, but your son's obviously not going to be vaccinated for, hopefully, this summer, this fall. So how has that sort of affected the way that you, behave at work or in your community?
Yeah, I mean, we've definitely just reduced on everything, like going out in terms of, like, we used to go shopping once a week. Now we go once in 2 weeks. We just stock up more. I've also started getting a lot of home delivery services. So I have a compostable service that comes home and picks up my trash, delivers bread and eggs. I also have someone who comes and delivers milk. So just things like that. So a lot of it has gone online. And social life is, I would say, pretty much zero at the moment. But I mean, I have seen friends here and there occasionally, people you know and trust, and in very small settings, hopefully outdoors. I mean, in this winter, of course, things have to be indoor, but it has to be done really thought through with people and are safe, and committed to being healthy and safe during this pandemic. Basically, just do your best. That's about it.
Yeah. And then, so what's next for you? So we were discussing earlier, you're looking for jobs.
Yeah.
What do you sort of envision for your next step?
Yes. So I'm trying to develop a research program of my own. I'm really excited about that, actually. So I think my goal, at least for now, is to develop a program which looks at advantageous microbes and commensals in training your immune responses and preventing that pathology that you get from different diseases, be it tumor or an infectious agent, and just break down into projects, get grad students. I mean, that's a very far off hope. I'm just interviewing right now, but hopefully I get one and then I move.
Yeah. All right. Well, thanks for talking to us. Good luck.
Thank you. Nice seeing you again.
Sharmila hopes to establish her own lab soon, focusing initially on how adventitious and commensal microbes train host immunity and modulate pathology following infection. This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, Spotify, and other podcasts, or at lmtv.podbean.com. If you are a virologist interested in sharing who you are and what you do, please contact us at letusmeetthevirologists@gmail.com.